Okay, hi everyone. Let me just adjust a few things. Okay, um, here we are in the cell bio. Let's see. Oh, hi Lucy. Okay, so today is the cell bio. Feel free to ask any questions that you have, and I'll just start answering them. Otherwise, I guess I'll just start going through material. Okay, so let's see. Um, Since I don't see any questions yet, I'm going to go ahead and start talking about the biomolecules to start with, and then go from there. Okay. And like I said, just feel free to ask questions, and I will just answer them as we go. Okay, so DNA. Okay, so DNA is a code for proteins. So if you think about it, in order to produce all the stuff that's in a cell, have to be instructions of how to do that. And so essentially the DNA, which is made up of nucleic acids, is the instructions of how to make all the proteins and things that make up the cell. So that's why we often say like the DNA is the code because it's the instructions, right? And what scientists want to do is figure out how to read the code so that they understand how proteins are made in your body and living things. Let's see. So let's go over the biomolecules, and then if people have questions, we'll go through those. Okay. So we have four types of biomolecules. We have our lipids, we have our proteins, we have our carbohydrates. Sorry, I'm writing this down for you guys. And our nucleic acids. Okay. I don't know if you can read that. Is that backwards? It might be backwards, huh? Yeah, that's backwards. Okay, let's try something different then. So remember, in nearly every case, we have our monomers and our polymers. The monomer means that it's the small building block to the larger molecule. So for example, I have some paper clips here, right? Each individual paper clip is a monomer, right? It's the building block by which if I string them together, I can end up with a much larger molecule, right? In this case, it would be a chain of paper clips. Okay, so this is my polymer, whereas then my monomer is my single unit. For carbohydrates, the monomer is monosaccharides. An example of a monosaccharide would be glucose. And the polysaccharides are the polymers. So we have starch, which is found in uh, plants as their stored form of energy. We have glycogen, which is how we as animals store our energy. And then we have cellulose, which we find in the cell wall of plants. And then, so all of those are larger molecules that you put the monosaccharides together. Then we have our lipids. Now, lipids are the only one where you don't really have a defined monomer and polymer. You could say that the subunit is the fatty acids, but it's not really quite um, the, the same thing. Then we have our proteins. Um, in our proteins, we have amino acids. Here, Sam, you can say hi.
Hi.
They can't see you. Hold on.
Hey. Okay. Hi. Hi.
Okay, so and then we have our proteins. The monomers are amino acids. And then when we string together the amino acids, we have our polypeptide chain, which is our polymer. And then last, we have our amino acids. The nucleic acids. You do not need to know about disaccharides, no. Disaccharides, I mean, the only thing I want you to know is the idea that you put two monosaccharides together and you get a disaccharide. But that's the extent to which I need you to know what a disaccharide is, right? It's the idea of putting two monomers together to get a slightly more complicated molecule. But no, that's the extent of what I want you to know. That's it. Okay, Sammy, are you gonna go do your thing now?
I need help put on.
Oh, okay. Sorry. We will multitask here. Oops.
What does multitask mean?
That means you have to do more than one thing at once.
Just talk.
Okay, then we have our nucleic acids. And with nucleic acids, put your arms, please. Okay. Oh my, sorry guys, this is not expected. Okay, go. Bye-bye. Then we have our nucleic acids. With nucleic acids, you have DNA and RNA. Those are your polymers, and those are strung together by nucleotides. There are small differences between the nucleotides for DNA and RNA. With DNA, the sugar is deoxyribose, whereas with RNA, the sugar is ribose. And then the bases are different. We're going to get into that a lot more later on in the semester. So for now, just know that there are some differences. The only difference, though, in this case are the sugars. And then for the bases, they all have nitrogenous bases, which means a base with nitrogen in it. Yes, the CER is due on Sunday night, please, to turnitin.com. And then bring me a printed copy on Monday, please. And then, yes, DNA is usually double-stranded, and RNA is usually a single string of nucleotides. Okay, what else? What other questions do you guys have? Or what other topics would you want me to go over? Anything? Yes, okay, so hypertonic and hypotonic. Okay, so actually, Amaya's asking about mono and polysaccharides. Let me talk about that first since I was still on the biomolecules, and then I'll go to the hypo and hyper stuff. Okay, so monosaccharides are our monomers of carbohydrates. Polysaccharides are the polymers. That's the main difference. That is the difference, right? So essentially, you make a polysaccharide by putting together a bunch of monosaccharides. Okay, hypertonic and hypotonic. So first of all, remember that they are terms that are comparative. So you can't just say something is hypertonic or hypotonic unless it's in comparison to another solution. So if I have a cell, well, let me actually look at the review just to give you something concrete so we're all looking at the same thing, right? So when you look at the second to last page of your review sheet, right, it asks you about like different cells placed in different solutions, right? So what you're doing here, yes, I will explain protein structure next. So let me do the hyper and hypo stuff, and then I'll go to protein structure. So with hyper and hypertonic and isotonic for that matter, it's a comparison. So let's start with isotonic because I think that's the way to make it the easiest for you to see, right? With isotonic, and let me see, maybe my little demo will work if I don't write any words on it. Okay, so if I have a cell, right? Let's see, it's gonna work. Adjust it. You can kind of see that, right? Okay, so if I have a cell, and in that cell, there is a certain percentage of solutes, right? We'll just call it salt to make it easier, right? I'll make it the 3 dots there, and let's say I have the same amount. Oops. Okay, same amount in the outside solution. Then that means that it has the yes, Minsu, I will talk about the cell membrane too. So if we have equal parts of solute and solvent, right, in the cell and the surrounding solution, then we call them isotonic. What that means is that since they are balanced, then equal amounts of water are going to move back and forth, right? Yes, those of you who are asking more questions, don't worry. I promise I will get to your questions. No, thank you, Sammy. Okay, Sammy, go, please. Thank you. No, Sammy, you need to go, please. Thank you. Outside.
Mom, I just want to tell you something. I can't find pants for tomorrow.
Okay, we'll talk about it later. Thank you. Go outside. Thank you. Sorry for the interruption, guys. Okay, so isotonic then, remember that's dynamic equilibrium. That means that equal amounts of water are moving back and forth, right? Now, in comparison, let's say now we have a solution where there is a lot more solute inside, right, compared to outside. Now what we would say is the inside of the cell has more solute, less water, so that's hypertonic, whereas the outside compared to the inside would be hypotonic, okay? So hypo means that comparatively there is more water, less solute. Hyper means there's less water, more more solute, right? Now, how does that work in terms of then osmosis? Well, in this case, what we tend to do is we tend to say, okay, the solution that the cell is placed in, right? So in this case, because there is less solute and more water in the outside, then we would say that this outside is hypotonic, right? So if it's hypotonic, it means there's more water here and less water inside. So that means that water is actually going to move in because it goes from high to low. And if it does that, then it's going to make the cell swell. So if a cell is placed in a hypotonic solution, then the water will move from the outside in and it will swell. In comparison, now I'm going to draw a different picture, right? In comparison, now if I have more solute outside and less solute inside, now that means there's more water out inside than outside, right? So again, water moves from high to low. So now the water would move out. If the water's moving out now, that means I'm going to have the cell shrink. So when you put a cell in a hypertonic solution, the cell will shrink. If you put it in an isotonic solution, it will neither shrink nor swell because the equal amounts of water are going back and
Mommy,
forth.
can I trade with you?
Yes, you can take it. Thank you. Okay. All right, so let me go back and answer some of your other questions now. All right, so protein structure, that was the last question. So protein structure is interesting in that you have it all at once. Okay, Sammy, I need you to leave, please. Thank you. No, you need to go. Sammy, this is not okay. So let me find something to help you with that. Sorry, I'm trying to draw from what I've got here. All right, well, we had our chain of proteins, right? So you have primary structure, secondary structure, tertiary structure, and quaternary structure, right? So primary structure is just the order of the amino acids. So for example, if I have 20 different amino acids, right, then I might have a chain that goes in a certain order. Well, let's say my amino acids in this case are different color paper clips. In that case, my monomers are amino acids, right? And I have a chain here, which is going to be in a certain order. It's green, orange, blue, pink, green again. Okay, that's the primary structure. So the primary structure is just what order are the amino acids in. Now, what happens though is amino acids that are not next to each other start to form these patterns, what we call alpha helices and beta sheets. So sometimes they'll kind of twist around like a curly pasta. Um, and that's called an alpha helix, or they'll form like a beta pleated sheet. Like if you make those fans when you're little, my daughters like to make those, right? That's like a beta pleated sheet. So if it has that sort of pattern, and that's actually because of hydrogen bonds between the amino acids, then that's secondary structure. Now notice it still has primary structure, right? Like I can take this and like turn it into a curly cube, right? And it has that sort of shape, but it now still has the primary structure. I didn't change it. And then I make it into like a pleat, still has that structure, right? Tertiary structures now when the R groups of the amino acids basically have certain bonds they form and it gives it a very distinct shape. The shape of the protein gives it its structure, its function, I mean, okay? And then if I have two chains, so if I had like one chain of proteins and another chain of proteins and they were next to each other interacting, that would be quaternary structure. So, sorry, let me close the door for a second. So a protein will basically have all levels of protein structure. Well, I shouldn't say all. If it's one chain of proteins, like it's one chain of amino acids, it's going to have primary structure, secondary structure, tertiary structure all at the same time. It's not either or. And if you have two chains of proteins, then it actually has all four levels: primary, secondary, tertiary, and quaternary. Okay. Can I explain about the smooth and rough endoplasmic reticulum? Okay, so they're both organelles that I'm only going to find in a eukaryotic cell. The main difference between them is, well, their functions, first of all, right? The rough endoplasmic reticulum is where you are going to have ribosomes stuck to it. So the rough endoplasmic reticulum with those ribosomes is going to make, when the proteins get made, hi, Gina.
Hello.
Okay, thank you. Sorry, we have a lot of guests coming in today. Go, please. Okay, thank you. So when you have the rough endoplasmic reticulum, the idea is that a protein is made on the ribosome. The endoplasmic reticulum doesn't make the ribosome, right? Or doesn't make the protein. But then it will take that protein the ribosome made and it will modify it. It's modifying it so that it can get ready to go to the Golgi apparatus so that it can then be exported to leave the cell. So that's the rough endoplasmic reticulum. They call it rough because it has these ribosomes stuck to it, so it looks rough. The smooth endoplasmic reticulum, on the other hand, doesn't have anything to do with proteins. It makes lipids, so you could say then it plays a role in making the cell membrane, right? And then it also helps detoxify the cell. So if there's something poisonous that's in the cell, it's going to take that poison and break it down in chemical reactions. Okay, Minsu wants to know about the parts of the cell membrane. Okay, remember the cell membrane, the bulk of it is made up of phospholipids, right? And that's what we call a lipid bilayer, right? It's the two layers of phospholipids that are pointing towards each other. Okay.
Mama.
Can you guys talk to Daddy, please?
Mommy, is it okay if I take some?
No, I need this right now.
I need it.
Okay. Sorry, you guys. You guys really need to go talk to Daddy. You can draw in a second. Thank you. Okay, so this is the main phospholipid bilayer. Sammy, please go. Thank you. If you come back in 2 minutes, I'll give it to you. Okay? Thank you. Sorry guys, there's a lot of interruptions today. Okay, so this is a phospholipid bilayer. It's mainly made up of phospholipids, which means that the bulk of it is this. But then what we have stuck throughout are protein channels and things. Remember, the protein channels are what help the not the polar molecules go through, and sometimes ions as well. So anything that's not small and non-polar basically needs some help to get across the membrane. So you're going to have protein channels, protein transporters. You might have protein pumps in there. And then on the surface of your cell, you're also going to have like carbohydrate chains, and those help with like cell-to-cell recognition. So what they do is they call the cell membrane, they call it a fluid mosaic model, which is to say that it's fluid because it has fatty acids.
Mama, 2 minutes?
It hasn't been 2 minutes, please.
No. It has. Is it okay if I race?
Yes, you need to go outside now. Thank you. Okay. So then you have the fluidity is due to the fatty acids. If you have, and then cholesterol. So cholesterol and the fatty acids together make sure that the membrane isn't too liquidy, but not too solid either, because you want things to be able to move across. Let's see, what else? Can I explain hydrogen and covalent bonds and the overall main things to know about properties of water? Okay, so all of that is tied together, right? Covalent bonds are how atoms in molecules stick together. So water is our best, easiest example of that, right? When one oxygen and two hydrogens are stuck together, the oxygen is basically stuck to the two hydrogens. So let's just keep it simple. The oxygen and the hydrogen, that's a covalent bond, because in order to stick them together, they're sharing electrons, right? So that's inside of a molecule. Now, the thing is, they don't share the electrons equally. So oxygen basically pulls on the electrons more than the hydrogens. So because they do that and electrons are negative, the electrons are hanging out closer to oxygen than they are hydrogen, which makes the oxygen a little bit more negative and the hydrogen a little more positive. That's going to create what we call polarity, because now that means if you have another water molecule, I'll just keep it simple. It doesn't have to be water. It could be another polar thing, but just to keep it easy, right? You have another water nearby. Well, it's got the same situation. The oxygen's a little negative. The hydrogen's a little positive. Well, what that means then is opposites attract. The hydrogen on the first one is a little bit positive, and it's attracted to the oxygen on the other water, right? And then so that makes them form a hydrogen bond. So hydrogen bonds are between molecules. Covalent bonds are inside of molecules. Now, because water is polar, then water can form hydrogen bonds. So when water forms hydrogen bonds, then it gives it those sort of special properties, right? It gives it the properties of cohesion, adhesion, the ability to dissolve things, and the idea it's a universal solvent. So cohesion means water sticks to itself, right? Water will hydrogen bond to itself, and that's how you get what we call capillary action. It's how like water gets pulled all the way up, like a 300-foot tree, or even a 5-foot plant, right, or a 3-foot plant. In comparison, we have adhesion. It's the same idea as just when water is hydrogen bonding to other polar molecules, so it sticks to those other things. Okay. Um, let's see. There's the idea that water dissolves things. Well, only the things that are like it, right? So um, it won't dissolve non-polar things like oils, but it will dissolve polar molecules like glucose or allow ions and things like that to dissolve. And then water is a solvent, right? So we dissolve things in water. There's a question Svenja asked, do we need to draw the structures? You need to be able to recognize the biomolecule structures. I won't ask you to draw them. Okay, so, and Matthew's asking, why do cells use active transport? Like, why would they need to go from low to high concentration? So remember, diffusion is very much the idea that just wherever there's more of it, it'll flow to where there's less of it. It's a random movement. There's no little brain. There's no it doesn't know it's supposed to go anywhere, right? And that assumes then that that's okay. Well, the reality is in certain cells, there's going to be a situation where you need like a certain concentration, right? And so if you don't have that concentration and you're relying on diffusion to do it, that's not what's going to happen. So active transport is really important when you have a particular type of situation or cell where you need the concentration to be, let's say, a certain place. So you would use active transport to push molecules into a certain area where they maybe wouldn't naturally go due to diffusion. So that's sort of an idea. Like your neurons or the nerves of your nervous system are a really good example of that. Like they have to have a certain concentration of ions on either side. And so those ions have to be, in a certain concentration. So if they aren't, then the ion pumps will pump things across to get them there. Okay. Let's see. Microtubules and filaments, you don't need to know those. Essentially, those are just proteins that are in your cells. They have to do with giving your cell like structure.
Hi, mommy.
Okay. Hi, baby. Isotonic, Amanda says, isotonic is only used to facilitate diffusion, right? Since, no, isotonic just means if I'm comparing two situations, like two solutions, and they have the same concentration of water and solutes, then they are isotonic to each other. So yes, but normal osmosis, right? Osmosis is the diffusion of water. So they're related, but it's not that isotonic is only with facilitated diffusion or anything like that. Motor proteins are also just other proteins in the cell. They actually help things like move around. Cytoskeleton is protein. It's like a scaffolding in the protein, in the cell that gives it a certain shape. Do you need to know the different? You should have a general idea of what proteins can do. But if it's not something I've ever mentioned in class, I don't expect you to know it. If that doesn't make sense. Like I'm not going to ask you to find some really obscure function that we've barely mentioned. You should know, yes, basically the difference between DNA and RNA. Format of the exam is very similar to last time. It's a bunch of multiple choice. There's one A-level question, so one, more complex question, and then a few that are kind of a little more so the multiple choice questions, there's a variety, right? I mean, they could be like super basic to ones that require more, thought and putting things together. Okay. There's a question about enzymes and substrates. So enzymes and substrates. So enzymes are proteins, right? Remember, their function is to catalyze chemical or biological reactions. So when they catalyze reactions, what they're working on are the substrates. The substrates are the reactants. It could be one substrate that goes in and gets changed into a product. It could be more than one substrate that goes in and gets changed into a product. But in any case, it's somehow
Say hello.
in any case, it's going to react with that substrate to change it into something, right? Now, okay. So when we have a substrate, an enzyme working on a substrate, then that means it's going to make that reaction happen. Okay. The way that it makes that reaction happen faster is because it actually reduces the activation energy. It makes it easier for the chemical reaction to happen, right? And so in doing so, right, a reaction that would otherwise take a lot longer to happen occurs faster. Now, remember, there is a limit to that, which we call the saturation point, right? Like if I have like 32 enzymes in a test tube and I give them a whole bunch of substrate, well, initially you're going to see more and more substrate turning into product. But at some point, it's going to basically turn into a situation where every enzyme is busy. And when every enzyme is busy,
I need this, mommy.
okay, you take it. Okay, thank you. Oops, sorry.
I need this, mommy. I need this.
So when you have too many substrates, essentially, I shouldn't say too many, but basically there comes to a point where the rate of reaction, and that's measured by how fast the product is being formed, it levels off because every enzyme is busy. Then there's no way for it to go any faster than that. Yes, this stream will be posted later. Like as soon as I go offline, YouTube automatically like converts it to a video that you can watch whenever. Okay, let's see. So yes, you do need to know the basic differences between DNA and RNA. Do you need to know all the cell parts? Yeah, I mean, you need to know what all the different organelles do, right? Okay, can I explain enzyme concentration and substrate concentration? Okay, so concentration essentially is a measure of how many things you have in a given solution, right? So one way to think about it is actually like juice, right? If you have like concentrate, I don't know if you've ever gone to like the freezer section, you can buy like frozen concentrate orange juice, right? What that means is it's mostly like orange juice with very little water. So it's very concentrated because it's mostly the orange pulp and stuff and not a lot of juice, right? If you add water to that, you reduce the concentration because now, like in the total, there's now, more water and less juice than before. I mean, comparatively speaking, right? So with enzyme concentration, it means sort of like in a given space, how many enzymes do you have, right? So if you have 15 enzymes and then in another tube you have like 25 enzymes, and the enzyme concentration in the tube with 25 is higher than the tube with the 15. Now, substrate concentration is the same idea. It's just now we're looking at not how many enzymes I have, but how many substrates I have, right? So both play a role in sort of measuring how fast the product is formed. You just want to be careful not to mix them up. Okay. Let's see. We talked about the cytoskeleton. Let's see. What type of protein allows cells to communicate with other cells? Okay, that's actually a pretty complicated question. So essentially, there are proteins that are on the surface of your cells that aren't involved in transport. They're often essentially like a protein receptor. So there are proteins basically that are embedded in the membrane that don't transport things in the same way, right? So what happens in that case is when a chemical signal like a hormone comes and it sticks to that receptor, then it triggers a whole bunch of reactions to happen inside the cell that will then cause the cell to do something. So that's how they would communicate with other cells, because that first molecule that bound to the protein receptor probably came from another cell. Exocytosis is essentially when you have a molecule. It could be like protein or it could be anything else, or not anything else, but it could be a different molecule. But essentially, if that molecule needs to be exported or spit out of the cell, then one way that that might happen is it's in a vesicle. The vesicle then fuses and becomes part of the cell membrane. And then in doing so, it kind of just spits it out on the other side. And so the opposite of that would be endocytosis. Let's see. Okay, so the difference between DNA and RNA, someone asked earlier, or I talked about earlier, but I'll repeat it really quickly. So DNA and RNA, the main differences are the sugars and the bases. DNA has the sugar deoxyribose, RNA has the sugar ribose. They also have different nitrogen bases. They all have nitrogen bases, but they have different ones. So Some of the bases are the same, um, but the ones that are different is RNA has a base called uracil and DNA has a base called thymine. Um, the functions of polymers and biomolecules, that really depends on the biomolecule and the polymer, right? I mean, um, in the case of carbohydrates, the polymers are going to be varied in what they do. Glycogen and starch are for energy storage, whereas cellulose is for structure. In lipids, the polymers are going to be lots of different things, right? It could be something like um, a hormone, which is important for sending messages between cells, or it could be, you know, a a lipid that's important in like storing energy as well. Proteins have tons of different jobs, so you can't really say that there's one function. There's a lot of different ones. The main difference between protein channels, carriers, pumps, and transporters. Okay, so protein channel, they're all involved in moving things across the membrane. The big differences between them come in how they work, what they look like, I guess you could say, and what they're involved in. So a protein channel is like a tunnel, right? If I want to get from one end of like on one side of the cell to the other side, um, then and I just take it through a tunnel, that's a protein channel. Now, that being said, it's not a tunnel that everything can pass through. There are specific channels, like certain ions have certain channels, and the other ions can't go through them. A protein transporter is the same idea. So, protein channels use passive transport. A protein transporter also uses passive transport, but it's a little different. It's the idea that when you bind something to a protein, you change its shape, right? So in this case, if I have something bind to the protein transporter, right, and it changes its shape, it causes it to change and then it drops to the other side of the cell membrane. In doing so, it didn't cost any energy to do that because the simple act of binding that molecule to the protein transporter changed its shape and made it spit it out on the outside. So still passive transport. In comparison, the protein pump is going to be something that is active transport, right? Because that's actually going to move things from low to high concentration. And so protein pumps are involved in the active transport, whereas the channels and the transporters are involved in passive transport. The main difference between simple and facilitated diffusion is just that. Simple diffusion just means things go across the membrane, high to low. If they're small enough, they slip through the membrane and they have the right, chemical properties, like they can go across. On the other hand, with facilitated diffusion, that's where you have to travel through something. It can't go across the membrane by itself, so it needs to go through a protein channel or a protein transporter. Let's see. How can cellular respiration be connected through cellular transport? I'm not sure what you quite mean with that. Cellular respiration is something we're going to talk about in a lot more detail in the next 2 weeks. But simply put, cellular respiration does require energy, right? I mean, sorry, it's the production of energy, and you're going to make different molecules, right? So, simply put, part of it might be, well, let's put it simplest, right? Glucose is what you use in cellular respiration, right? And so that's going to happen in the mitochondria. When you eat, you have to get the glucose that you eat into your cell and then into your mitochondria, right? So cellular transport could be involved in that sense, in that first you have to get the glucose across the membrane into your cells. So that's going to happen through facilitated diffusion, right? And then it would even need to go into, well, actually, the glucose itself doesn't need to go into the mitochondria, but the idea is that molecules might have to pass as well in order for cellular respiration to happen. So I guess that would be how you'd connect cellular respiration and cellular transport, in that you have to actually um, take the glucose into the cell, right? And the way you do that in this case would be facilitated diffusion. Motor proteins actually just help proteins move through the cell. Think of them sort of like a subway system. We didn't get into that, so I wouldn't worry about that. Glycoproteins are those carbohydrate chains on the surface of cells. They help with like cell to cell recognition. Glycogen, on the other hand, is very different. Glycogen is the polysaccharide that we use in our liver and our muscles to store energy. Glucose is not the only type of monomer in carbohydrates. It's just one we talk about a lot. Um, Anything where it's like basically what we call a monosaccharide. So fructose, glucose, galactose, those are all monomers. The definition of a monosaccharide is just sort of that single ring of a carbohydrate. Uh, no, for me, I can't guarantee other teachers if anyone who's not my student is watching. For me, I don't really need to know anything too defined about RNA and DNA except the basics, which is what I told you in class. Yes, endocytosis and exocytosis do require energy. Can I show the diagram on page 1 of the review packet for enzymes? What diagram is that? I'm not sure. Yes. Okay, so Ranger will, yes, polysaccharides would have multiple sugar molecules, yes, while monosaccharides only one. That's correct. Yeah, Arul, I'm not sure what you mean by the diagram on page 1 of the review packet. I don't see an enzyme packet diagram. I don't see it. So if you can clarify, that would be good. Okay, anything else? Let's see. It's at the bottom. Do you mean the review packet that I gave you? Let's see. Do you mean... Oops, this is not going to work. Let's see if we turn off the light if that helps. Is it this one? Yes, okay. So what is your question about that? So basically, if we're talking about draw a picture of reaction, it's just the idea that you want to show a substrate going into the enzyme and causing a product, basically. When would a cell, okay, so Joshua asked, when would the cell need to use active transport? Again, it would use active transport when it needs to have a certain concentration of molecules, and diffusion's not going to get it there, right? Because diffusion basically just is random movement until things are equal. But if I actually need for a cell to have, let's say, more sodium on one side than on the other side, or more potassium, then it's often going to have to pump it in or pump it out in order to do that. Arul, what exactly is your question about the enzyme diagram? Okay, what is the difference between a monomer and subunit? Well, I mean, technically they're different things, right? I mean, but I guess we're not really differentiating too much about it. We just call it for lipids like a subunit as opposed to monomer because we don't really have a defined like unit that we could string together into many to make a chain. So I wouldn't worry about that. Compare exocytosis and endocytosis. So they both involve vesicles, right? One essentially they're opposites. You can think of it that way. Endocytosis is how the cell would take something in. So you would have a molecule that comes up to the cell membrane. The cell membrane basically kind of forms like almost like a C around it, right? And then it would sort of go in and it would just close up and go into the cell membrane and then sort of get pinched into the membrane. Whereas with exocytosis, it's the opposite. You have this like bubble of, let's say waste or protein, right? It's going to actually fuse and become part of the cell membrane. When it does that, it kind of spits it out on the other side. But in both cases, they require energy, and they both also use vesicles to do that. Let's see. Joshua says, so is molecule movement hypotonic solution and active transport? I'm not sure what you mean by that. So if you could clarify that, I'll come back to that one. Cohesion and adhesion is basically the idea of properties of water. Properties of water are due to hydrogen bonds, right? And so because water has hydrogen bonds with other water molecules, it allows water to stick to itself. And it also allows water to stick to when I say stick, I mean hydrogen bond to other polar things. Is there anything on the test that is not in the Unit 2 review? Remember, the review is just a general, like, I can't guarantee that, like, conceptually, no, there's not anything else out there, right? But the questions could bring something that you're not familiar with and give you a like a context of it and ask you to apply it. So in that sense, like, it's not going to be like word for word what's in your exam. Um okay, so let's see. What makes water polar? What makes water polar is the fact that oxygen and hydrogen basically pull, when they're sharing electrons, they don't share equally. And anytime you have electrons not being shared equally, then they are basically going to create a situation where the electrons hang out closer to one atom compared to the other one. And then that gives that atom a slightly negative charge. And then the other one a slightly positive charge. So that's what makes it polar. Um, what about size and scale should you review? You don't need to know specific sizes of things, right? I think you just want to kind of keep in mind the general idea of how size and scale play into a lot of the things we're talking about, right? For example, like diffusion. Um, Molecules are going to only be able to cross the cell membrane or any semi permeable membrane depending on their size. So there are going to be molecules that simply can't get across the membrane, not because the membrane has anything against them, but because they might, there are essentially microscopic openings in the membrane. And if the microscopic opening is too small for the molecule, it can't cross. I would say that's sort of the main thing. Sahana's asking me to go over one of the osmosis practice problems. Okay. Um, why don't I do the one that's on your review sheet just for context? I don't actually have the other ones with me. So hopefully that helps. Remember, in this case, what I would really do in this case is, okay, I see your question, Joshua. I will get to that in just a minute. So with any of these osmosis questions, what I would say you should do, like personally, what I do is it's hard for me to keep track sometimes of like where things are, right? So if you draw a picture of it, that's going to help. Like I would literally draw the solution and the cell or with the membranes, the two sides, and that way you can see where everything is. Because what you want to figure out is where is there more water and where is there less water. So, for example, it says a cell is 95% water is placed in a 10% glucose solution. So the first thing you need to do is actually figure out what does that all mean in terms of water, right? So the cell is 95% water. That means it's 5% other stuff. I'm not even going to worry about what the other stuff is because I really, in this case, want to worry about the water. It's placed in a excuse me 10% glucose solution. Sorry. So if it's a 10% glucose solution, that means it's 90% water, right? So that means on the inside of the cell, it's 95% water. And in the outside of the cell, it's 90% water. Those are the numbers that matter in this case for osmosis, because that's going to tell me what direction water is going to go. So since there's actually more water, a higher percentage of water inside the cell compared to outside the cell, then water will leave the cell, right? It would flow out. If it flows out, then my cell is going to shrink. And what it means is that my solution outside of my cell had less water, more solutes. So that makes it hypertonic to the solution inside. Let's see. Hopefully, Sahana, that helps. Okay. If you still have questions, let me know. Okay, so let's see. Josh was asking if a hypotonic solution, when they start moving out of the cell, does it require energy? No, I mean, if we're talking hypertonic and hypotonic, we're really just talking about the movement of water, in which case it's diffusion or possibly facilitated diffusion, but in either case, it's all passive, so it doesn't require energy. What do hormones do and what type of biomolecules are they? Hormones are typically one of two types of biomolecules. Some of them are lipids. Anything like a steroid is a lipid. So, like cholesterol, well, actually, that's not a hormone. Testosterone, estrogen, those are all steroid hormones. But then you have other hormones that are proteins. So, basically, hormones are split. Some of them are proteins and some of them are lipids. Why does a cell absorb dead cells? Well, it probably depends on the situation. There are some cells whose job it is, like immune system cells, to kind of clean things up. So cells don't, in general, like take in other dead cells. Yes, semi-permeable and selectively permeable are the same thing. What happens when amylase breaks down starch? Good question. So amylase is a enzyme. Its substrate is starch. Remember, starch is a polymer of carbohydrates. So what it is essentially is glucose strung together, or monosaccharides strung together, right? So when amylase breaks down starch, what it actually does is it breaks those monomers off of the starch. Like it breaks apart the starch into monomers. That's why you actually have amylase in your spit. So if you take like a cracker, which has a lot of starch, and you hold it in your mouth for a while, what's actually going to happen is it's going to start to taste sweet. The reason it starts to taste sweet is your enzymes in your mouth are actually breaking down the starch in the cracker into glucose, and then you taste it because you have receptors for that. What is an example of a possible evil question? You want to be able to put things together, right? So you want to be able to, for example, describe topics that go together. Like, could you describe, um, aspects of biomolecules and cell transport, or could you describe things like cell transport and water movement, or mixing and matching all those different terms together? Uh, Facilitated diffusion does not require energy. No, it is considered passive transport. Can I go over the functions of the different organelles? Um, I would say it's all in the interest, I guess, of going faster. I'm not sure if me repeating it to you is necessarily going to help. I guess my question would be: is there a specific question you have about a particular organelle? Because I feel like otherwise I'm just going to read to you more or less what's on the review sheet. Just remember, every organelle has like its own job, right? So mitochondria are involved in energy production. Um, the ER, so you want to be able to tell me which ones are involved in protein production, right? That's going to be like the ribosomes, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, they digest things. So they all have different roles. Well, we have to draw a diagram that shows how proteins are formed. No, I mean, I want you to understand about proteins, but I don't need you to know that specifically. Yes, facilitated diffusion needs the help of a protein in the cell. Remember, that's the whole idea that things can't cross the membrane. Certain things can't because maybe they're polar or too large or both. So I need to do that. Bulk flow is just the idea that when you have like, uh, it kind of is idea of pressure flow. So it goes from like high to low pressure. When you have a lot of water, it creates what we call osmotic pressure, and the water will flow from where it's higher, more of it to less of it. So it's related to, um, the ideas of osmosis and such. In terms of knowing where each organelle is, no, like it's not like I need to tell you to tell me, like, for that matter, it doesn't even, everything doesn't just stay in one place in the cell, right? I want you to be able to tell me what an organelle is and where what it does, be able to recognize it based on its functions and things like that. But it's not that I wouldn't ask you, like, it's not like there's one picture that's always where something is in a cell in terms of organelles. Cellulose is a carbohydrate. It is a polysaccharide. The nuclear envelope is just another name for basically the membrane around a nucleus. So remember, it's not that prokaryotes don't have organelles. They don't have a lot of organelles, but they do have some, right? They have a cell membrane, they have cytoplasm, and they do have ribosomes, but ribosomes don't have membranes. So what you want to think about it is what the eukaryote has is what we call membrane-bound organelles. The idea that you can have all these different pockets and spaces in a eukaryotic cell where separate things can happen. And one of those really important places is the nucleus. So the wall that sort of creates the nucleus, that's the nuclear envelope. Let's see. Can I explain how an enzyme gets to its saturation point and what happens after it reaches its saturation point? Yeah. So initially, when you have an enzyme, if it doesn't have very much substrate, right, it's going to be able to produce product fairly, it's going to continue to increase its rate of production of product. Like the more substrate you add, the faster it's going to make produce product. But at some point, it's going to max out, meaning that at some point you're going to have all your enzymes busy, right? And if all your enzymes are busy, then the rate of reaction, which is how fast the product is being produced, basically levels off. It's not that it drops, it's just that it stays the same. And so that's how it reaches saturation point when it gets to a certain concentration where all the enzymes are busy and it's not going to make any product any faster, right? Kind of like if we were, let's say we were decorating cupcakes for a party, right? And each of you is like an enzyme decorating a cupcake, right? Well, if every single one of you is decorating a cupcake, then we've reached our saturation point. At that point, if I brought 50 more cupcakes into the room, you're not going to produce cupcakes any faster because you're all busy doing what you're doing. Um, what's the path of, okay, osmosis and diffusion? Diffusion is the general idea of thing moving from a high to low concentration. Osmosis is the diffusion specifically of water. The path of a protein in a cell is the protein is made in the ribosome, and from the ribosome, assuming it's a protein is going to leave the cell, which is what you normally then have that pathway for, um, then it's going to go from the ribosome to the rough ER, from the rough ER to the Golgi. Then it would actually go in a vesicle to the cell membrane and then leave. Um, let's see. On the last page of the unit review, there was an identifying organelles, and I don't really know which one is which, so could you show me it? Okay. That one's honestly, think more about in that case, like the overall functions. So, as a general rule, the nucleus is usually in the middle. So, let's see, I don't know if you guys are going to be able to see this. I'll try. Yeah, okay, kind of. Okay, so I'm going to try to point things out to you guys as best I can. All right. Let's see. This is the nucleus, right? This is the nucleolus, which is in the nucleus. Then here we have our rough endoplasmic reticulum. You can tell it's rough because it has those little dots on it. That's the ribosomes. This would also be rough endoplasmic reticulum, but notice here there's no dots. That's the smooth endoplasmic reticulum. This is most likely the Golgi. Looks kind of similar, but it's a little more sort of removed from it all. This is the mitochondria, kind of has this sort of jelly bean like shape. Honestly, in this picture, it would be hard to identify a vacuole or a vesicle, but it would look sort of like these little bubbles, you know? And then you've got cytoplasm and the cell membrane. Those are probably the easiest ones. No, I'm not going to ask you to know where every organelle is. I'm more concerned that you tell me what they do. But you know very general idea. Like I said, like typically the nucleus is towards the center, or if you have a plant cell, usually you have like a bigger vacuole in the middle where it stores more water. Um, let's see. How does insulin and glucagon help with blood glucose regulation? A very good question, but I'm not sure it's relevant to this particular exam. Simply put, insulin and it's glycogen or glucagon. I'm not sure what you mean. Insulin basically is the hormone that helps your cells take in um, glucose. So think of it as like they're the key that allow glucose to enter your cell through facilitated fusion. Um, and then glycogen is stored glucose. So that's also going to be important. Can I explain how being saturated or unsaturated affects bonds, being double and single? Being saturated, unsaturated, it's not that it affects bonds, it's defined by the bonds. So, especially in this case, we're talking about saturated and unsaturated fatty acids. If it's a saturated fatty acid, it's single bonds between the carbons. So, what I remember is SSS, single saturated solid. If it's a single saturated fatty acid, it has single bonds. It's going to be solid at room temperature. If it's unsaturated, then it's going to be have double bonds or triple bonds, right? Which means it's then going to be liquid at room temperature. Let's see. Do we need to know the polarity of different molecules? No, I mean, know that water is a polar molecule, glucose is a polar molecule. Um, me personally, I'm not going to ask you even that complicated of stuff. Can you explain monomers versus polymers and what they're used for in plant or animal cells? Okay, so again, monomer and polymer is a very generic term that simply describes the smallest subunit, and then when you string them together, that's a polymer, right? So what they're used for in plant and animal cells is tons of things. Um, you know, for carbohydrates, for example, a monomer is the glucose that you're going to put through cellular respiration, but then you store that um, in animal cells as glycogen and you store it in plant cells as starch, right? Um, in in the case of DNA, right, your polymers would be like nucleic acids and they're made up of nucleotides. And so you are going to have nucleotides that are floating around in your cell that you would use to make more DNA and RNA. Same thing with proteins, right? You'll have amino acids that you use to make proteins, and proteins do tons of different things. Other than the nucleus, what is the difference between prokaryote and eukaryotic cells? It's not just the nucleus. Remember, it's the idea that a prokaryotic cell has only ribosomes and a cell, cell membrane and cytoplasm. It doesn't have what we call membrane-bound organelles. It doesn't have separate compartments inside of the cell where different things can happen. Whereas in the case of a eukaryotic cell, it does have all these different organelles that have all these different compartments, right? So mitochondria has a membrane around it, the nucleus has a membrane around it. Um, so I think you want to be careful not to just say that. That's what people say a lot, like, oh, the nucleus is a prokaryotic cell doesn't have a nucleus. Well, you're right, it doesn't have a nucleus, but it also doesn't have other membrane bound organelles. Why would a cell perform endo or exocytosis? Um, Essentially, it would do that to either get rid of waste or take in like food particles, right? You can kind of think of endocytosis as the cell's way of eating and drinking. So it would, take in food molecules and liquid molecules, and then it would, release waste. Or it could also be doing it to release a protein, right? When we talked about the path of a protein, like the path of the protein is that it needs to leave the cell. So when it leaves by exocytosis. Okay, why does the cell structure of a plasma membrane allow to be selectively permeable? Well, remember it's that lipid bilayer, right? And so that double lipid bilayer basically makes it so that, remember, that the fatty acid tails of the phospholipids are nonpolar. So something that is polar can't really get across. Something that's too big can't get across, right? And so then that's making it selectively permeable because not everything can now cross your membrane. You basically have to be small and nonpolar to cross the membrane unless you have some help. At the top of our review sheet, we are asked to identify different kinds of cells. Do you mean like prokaryote versus eukaryotic? I assume that's what you mean. And in fact, I mean, so in that case, I just want you to be able to do, um, just that. Do you need to know examples of adhesion, cohesion, and solvents? Not real. I mean, adhesion and cohesion, there's not really an example to it, right? Like cohesion is water sticking to water. That's all there is to it. With adhesion, it's water sticking to other things, other polar things. Past that, I don't really expect you to know an example. No, a solvent, yes, is anything you can dissolve a solute in. So water is what we consider the universal solvent, but anything could be a solvent, right? I mean, there are non-polar solvents, like very different from water. Let's see. I think you would see the image provided. Okay. So, Amaya, are you referring to the different biomolecules or the different kinds of cells? Um, oh, I'm sorry. I didn't mean to skip your R group question. Yes. So, with an amino acid, basically, amino acids are the monomers of proteins, right? And so they all have a lot in common because they're all the monomers of proteins, 20 different amino acids. And yet at the same time, there has to be something that makes them different. Otherwise, why would we have 20 different ones? So the R group is what makes them different because every amino acid has an amino group, a carboxyl group, and a hydrogen. The fourth element is that R group. Some of them are going to have just a hydrogen in the R group. Some of them are going to have oxygen and hydrogen. Some are going to have some sulfur in there, right? So that R group is really what makes that amino acid unique, because that's going to give it certain properties that make it do certain chemical reactions. So the way I told my class to think about it is, think of all the amino acids together as like a sports team, right? Like the Giants, San Francisco Giants, right? They're all a team, so they all have the same uniform, right? The same colors, the same you know, design, etc. So that's like the amino group, the carboxyl group, the hydrogen. But then each player has certain parts of their uniform that makes it unique, right? And that's their number and their name. So the R group is like their number and their name. Um, let's see. Amaya, you said a microtubule is just a type of protein. Um, you don't need to know anything else about it at this point. Let's see. I gave you the bond maps where I gave us the letters, example CHO, things like that, and told identify it. Is it the top of the Unit 2 review, ma'am? Essentially, all I want you to be able to do with the biomolecules is to be able to look at the picture and recognize which one it is and be able to tell me what kind of biomolecule it is, and then from that, maybe the monomer and the polymer of that biomolecule. What's the difference between motor proteins and regular proteins? Um, Motor proteins are a type of protein. There's no such thing as a regular protein. Like, proteins are just one biomolecule. So a motor protein is just a type of protein that allows things to move through the cell. That's sort of what they do. So they're defined by their function, right? But it's not that they're irregular proteins. What determines protein's shape? What determines a protein's shape is actually the order of the amino acids. That ultimately, it's complicated, but basically, ultimately, that determines how the different amino acids like interact with each other, and that then gives them a certain shape, and that gives them their function. No, you do not need to know CHIPO. That was from our last exam, so don't worry about that. Polarity, again, is the fact that electrons are not equally shared between atoms sometimes, and so when they're sort of tugged more towards one atom than the other, that's polarity. The main parts of the cell membrane, I did already talk about that, but I will go through it really quickly. But just to warn you guys, I probably have to go in the next 5 minutes or so. Okay, so the main parts of the cell membrane, again, the very bulk of the membrane is the lipid bilayer, right? The two layers of phospholipids. That is what makes, and the phospholipid head is polar, and the tails are nonpolar, and the nonpolar tails are facing towards each other. So that forms the bulk of the cell membrane, right? But then you have sort of embedded in there your proteins, your protein channels, your protein transporters, and your protein pumps. And then on the outside, you would have your carbohydrate glycoprotein chains, which are going to determine or help cells communicate with each other, or recognize each other. Any other questions? Last questions before I have to go? Remember, if you do think of something, you have a few options. Mr. Jones is probably going to be streaming very soon if he's not already online, and Mr. H will be on at 9 o'clock, or yeah, 9 o'clock. What do amino acids do? They're the building blocks of proteins, right? And proteins do so many things. So without amino acids, you would have no proteins. Remember, also, you guys can email me tonight. Like, if it's after, get some sleep for sure, right? Don't be up super late. But if you have questions, you have options. Like I said, you can come to see me tomorrow morning, or you can email me tonight, and I will check my email and I'll write back to you. Our groups are the things that make amino acids unique, right? Like I said, it's like the player's name and number on their back. Yeah. Okay. Anything else? Okay. like I said, email me if you've got questions. Don't worry. I think you guys will be fine. But um, do the review. I would say try to do the review and like quiz yourself. Like ask yourself, ask someone to ask you questions. Don't read your notes passively. It's everyone, me included. Like if you read your notes and you're just like, okay, I guess I get it, you're not really testing yourself, right? Think of it this way. Some you guys are gonna be driving soon, right? You wouldn't read a book on how to drive a car and then go take your driving test. You would get behind the wheel and actually start driving the car first, right? In practice. So do the same thing here. Um, that'll do better. You know, you'll have hopefully better results from that. So yeah, quiz each other, help each other out. Um, okay, I think I better go. Oh, and sorry about all the many, many interruptions from my children. Um, we'll do a better job keeping them entertained next time. Okay, have a good night, everyone. Get some sleep, eat a good breakfast in the morning, and I'll see you guys tomorrow. Okay, I'm glad you enjoyed their antics. All right, bye bye, guys. Have a good night.